Hello and welcome to the Neuron Regeneration Podcast. Today's episode is an interview with Dr. Reggie Edgerton, who is a huge name in spinal cord injury research for many decades, as well as neuromodulation, which I'll give you a definition for right away. The International Neuromodulation Society defines as the alteration of nerve activity through targeted delivery of a stimulus, such as electrical stimulation or chemical agents, to specific neurological sites in the body. And as you'll hear in our conversation, Dr. Edgerton and his lab have also been applying neuromodulation to cerebral palsy, Parkinson's, and other neurodegenerative and trauma contexts that I'm sure anyone interested in this topic of neuron regeneration will be very excited to hear more about. Let me quickly read a short bio from his lab webpage at UCLA, and we will dig in. Dr. Edgerton received his PhD in exercise physiology from Michigan State, his master's from University of Iowa, and his BS from East Carolina University. He is the director of the Neuromuscular Research Laboratory and a distinguished professor of the departments of integrative biology and physiology, neurobiology, and neurosurgery at UCLA. He has been teaching and conducting research at UCLA for over 40 years. His research is focused on how the neural networks in the lumbar spinal cord of mammals, including humans, regain control of standing, stepping, and voluntary control of fine movements after paralysis, and how can these motor functions be modified by chronically imposing activity-dependent interventions after spinal cord injury. And as I just mentioned, and as you'll hear, he is also working out of the Rancho Research Institute, and they are applying these neuromodulation interventions to, far more than just spinal cord injury. And yeah, really, really exciting stuff that we get into. So with all that said, thank you as always for joining us. And here is Dr. Reggie Edgerton. Okay, thank you so much, Dr. Edgerton. It is such an honor to have you on the Neuron Regeneration Podcast, and I appreciate you taking the time.
Thank you. Thank you for the opportunity.
Yeah, absolutely. So I've been following your work for years, as you know, and I think a great place to start for the audience would be to just get a good definition of neuromodulation, and then we can dive in from there.
Yeah, I think the term neuromodulation has emerged amazingly rapidly, and that has resulted in the term being used widely for different purposes.
Mhm.
And I think in general, you could think of neuromodulation as a way to shape the physiological state of the different neural networks that are involved in a given function. So in any task, the motor and sensory side, which both, of course, are essential, you can have a given sensory information that goes into networks and can get a response, but the response is going to depend on what the excitability or generally the physiological state of the nervous system. So, for example, when you get up in the morning, the physiological state of your nervous system is not the same as it is midday or after you've had a big meal or after many things. It's changing constantly. So a given stimulus will have a different effect depending on the physiological state of your nervous system. That's the bottom line. And we have all of these sensory systems, which are generally thought of as individual sensory modalities. And that's the way they have generally been studied. The limitation of what we've learned so far is the fact that they've been studied by an individual will spend their whole life studying how you hear or how you see. But there's been very little, primarily because of limitations in technology, of how these different sensory modalities are integrated. And basically, how you respond to any stimulus is not due to vision specifically or audio signals and so forth. It's really the result of how your nervous system has integrated all of these sensory modalities to give a final state. at any given time of what your physiological states are of the nervous system. So we need to know more about how these sensory modalities are integrated and how we can use that to an advantage to change the input to the nervous system that is going to give a certain response. And of course, we're thinking of this in a situation where the nervous system has been modulated in a way by a disease or disorder of some kind. And can we compensate for it? Because we know that you can lose sight and still walk. And so forth. So we have many examples of this. You don't have to be able to hear, but there are other ways that the system is able to reorganize itself so that it can compensate and figure out new solutions with what's left. And that has generally been my central question for over 50 years. And after having studied central pattern generation and seeing the automaticity and the complexity of that automaticity located only in the spinal cord, and when you're considering gait, that the question became, to what extent can we take advantage of that automaticity that may be remaining in many of the spinal cord injuries, to be specific, and this is where we started and spent most of our time. So how much recovery can we get with a system that remains, and how much can we induce some type of adaptation so it can function without those networks that it has lost? So the general scheme again is to better understand how our nervous system is using all of our sensory modalities to give us given response. And so in the process of doing that, we've learned a lot of basic biology that has generally not been known or at least been ignored. And I would make one other point here right at the beginning, is in this whole process, almost everything that we have observed so far in regaining the functions, it's all been from concepts that evolved from systems biology, not molecular biology. There's not a single critical observation in molecular biology that has led to any of the important concepts that have evolved. That is not to say at all that molecular biology is unimportant, but what it emphasizes to me in looking back on this is how rapidly our research system in academia and so forth keeps diving into the experiments that is more and more reduction. And as soon as you reduce the complication of the whatever you're studying, you're minimizing how relevant it is to the whole organism. And so we've left behind a lot of important systems biology concepts. And I've been trained as a systems biologist, basically, and have not had the time and capability to get into the molecular biology. Of course, I'd love to know all that, but I think I'm been challenged with learning as much systems biology and how it can be reorganized and adapt to remarkably adverse situations.
So I'd like to maybe loop back to the systems biology component because it reminds me a lot of someone else that I interviewed on this podcast, Dr. Michael Levin, and how he's using, we wouldn't call it neuromodulation, but rather bioelectricity. So they're not using electrical stimulation, but rather ion channel modulators to manipulate
Right.
patterns. And he often says we're working on the software of the system, not on the hardware of the individual wirings or cells or what have you. So it kind of reminds me of that. But if we stay on the definition of neuromodulation, can you take us back to when you or whenever it first started or was recognized or starting the experiments on actually imparting electrical stimulation into the spinal cord of someone who's had an injury there, and what were the kind of original results that compelled you to take it further for decades?
Well, it's difficult to state all of those, but I really followed the concept that if you start with a basic question, then you follow the results. I was not learning how much could not be done with spinal cord injury, for example. I was just seeing this, and I was trying to understand, and the basic question in my whole career has been, how do we move as we move? How do we control our movement? And so that in studying that particular question, have not been biased toward, well, no, spinal cord injury, there's nothing you can do about it. It's too tough of a problem. In fact, I've heard some scientists say no good scientist would tackle spinal cord injury. It's not good for your career. So, but I was not fed that kind of concept, and I just have followed the science. And the science has led me here. And the critical experiment that got me started in this direction was in the first study that I was involved in with Sten Grillner in Sweden, where we studied the concept of central pattern generation, which was just emerging very rapidly in his lab during the early and mid 70s. And when and our first experiment was, he had done more or less the organism systems level studies that showed the potential here, that if you eliminated the input to the lumbosacral spinal cord, these animals could regain the ability to step. And that an important observation, but this was done in animals that where the spinal cord was transected completely at a neonatal age.
Mm.
And everyone knows that in many conditions, the neonatal period is a critical period of adaptation and learning and developing. So the critical question, could this occur in adult? And after I left Sweden, and I had already decided this is the direction I'm going, and because the central pattern generation, what we were observing physiologically was just incredibly amazing. And basically, what it was showing us that of all of the muscles of the hind limbs, which were basically equivalent to what you see when you're walking. So that means that there were control systems already built into the spinal cord to cycle over and over in a way that activated the motor pools in a specific way that would generate these movements. But of course, if you've got all the nerves cut, you're not seeing movement, but you're seeing output to the nerves, motor nerves, that would generate the muscle movement. So, but the point is, you've eliminated all proprioception and all rhythmic input to the network, but it still looked like it was stepping. It was referred to as fictive locomotion. So that emphasized the importance of the automaticity that's built into the system. And it kind of initially kind of minimized my thoughts. Well, how important is the sensory information? But today, I know now just how critical it is and how much we're dependent on it, and as a way of inducing the adaptations that we need to occur.
And just
So,
curious with those experiments and since are we talking about providing an electrical stimulus below the level of the lesion, and then it's activating the central pattern generators that reside at all parts of the spine, or at that part of
Yeah,
the spine?
what you could do is we pharmacologically neuromodulated, and that's the other thing that I didn't get to earlier. You can electrically neuromodulate, you can chemically neuromodulate. So I think right now we need to be thinking of what are the type of input and signals that we can use to change the physiological state of the networks. It can be different types of electrical stimulus, it can be different waveforms, it can be a number of things.
Mhm. What kind of chemical stimulation causes the
Well,
modulation?
there were basically serotonergic type of drugs.
Mhm.
And we know that over years, we know that they're important. In fact, just in recent studies that we've done, we had already shown the effectiveness of serotonergic type of drug, buspirone, in rats. But in our recent human work, we've shown that it also effectively neuromodulates the system so it can facilitate neuromotor recovery.
Wow. So you're infusing that type of chemical in the area, and it's very similar to actually putting an electrical current into the area.
Right. In fact, in the animal experiments before we got to the human studies, and be brave enough to try this in humans, it took about 35 years before we got to the human. But we finally got there, and it was known even earlier that you could electrically and pharmacologically activate this central pattern generation phenomenon. And so, and what we found out eventually, when we were training the animal, we knew that training, it was not just the neuromodulation that is important. After you neuromodulate, you change the physiological state and the properties of those networks, but you haven't told it what to do.
Mhm.
If it's just sitting there plastic, how's it going to adapt? And if you don't give it any information to tell it how to adapt, then you don't get the full result that is possible. And that's where the training comes in, and that's where the sensory information comes in, because the sensory information, these sensory ensembles go into these central pattern generators. That's how the central pattern generator recognizes the sensory ensembles and knows exactly what to do next based on the sensory information that it gets.
And an example of that would be like the proprioceptive input of the weight on the foot or the position of the knee joint.
Absolutely.
All those
Ev-
signals coming from the bottom up to that CPG,
yeah.
it needs that in order to kind of kick things going or get things going and actually create movement.
Is critical.
Okay.
Is critical. And I think of proprioception as being all kinds of proprioception. The other thing that limited a lot of thinking in this area was the concentration on a specific sensory receptor, a spindle, a Golgi tendon organ, and so forth. And I think the most effective way to think of this is no individual sensory receptor is doing it alone. What the spinal cord is receiving these sensory ensembles that includes all the sensory receptors that are telling the body exactly where your foot or leg and is at any given time. It knows where it just was a few milliseconds earlier. It knows what it just did, and it already has told the nervous system what it's going to do next. And that's why you can get this continuous loop of complete locomotor cycle, step cycle, going for hours.
And if there's a full complete transection, so no input coming from the top down, you can, whether chemically or electrically, provide this neuromodulation, and it will still just the proprioception up to that part of the spine below the transection is enough to actually spark a gait pattern that will go continually.
So the electrical stimulation, the pharmacological stimulation, and the animal studies, by the way, we always got the best result with the best combination of pharmacological and electrical neuromodulation. So, because they're using slightly different mechanisms to enhance the functionality of the networks.
And how has it gone? Like, can you tell us the story of taking it into humans? And, obviously, most people who are interested in this topic, they've seen a lot of the stories about people getting whether internal or external electrical devices, helping them to walk and stand. And can you tell us kind of an overview or summary of when that started and how it's progressed since then?
Well, after the initial experiments in Sweden, and when I returned from my sabbatical at that point, our first question is what was being observed in the neonatal animals. In other words, they were injured in the first 2 weeks of the animal. That they had recovered quite a bit of the walking capability. So the big question is, in an adult, would the same thing be true? And in our first experiments, we found out this is an example where the data just told us what to do next, and we found out that the same thing would happen in the adult. Not as easily and quickly, but the same thing would occur using just training. At that point in the cat, it was not even necessary to use electrical stimulation. But eventually we got to the point in the cat where it's just too slow. We could only study relatively few animals in a given year. And so we said we got to go faster. So we went to the rat and mouse. And what we found out that what we were observing in the cat was not true in the rat and the mouse.
Hmm.
And so we said, okay, we're going to have to go to the stimulation. Like we knew in the earlier studies, you could also directly stimulate the spinal cord and get similar results. That had already been demonstrated. So then we started working on the technology to implant electrodes in the rat, and then we got into the pharmacology and the electrical stimulation there. So, and we've certainly found that we need the stimulation in the human subjects is very critical.
And then when was it that you started to take neuromodulation and do trials in humans, and how have the results or even the therapeutic approaches changed over time?
I would say in the around six, something like that, we were really getting to the point where we thought we were getting close to the human, being able to do the human subjects. And then, so we finally got a grant from NIH that let us to try it. And we knew that there was the technology that we thought would be good enough was already available, but it was used for pain suppression.
Mhm.
So it had been used, the technology was there, was already there, that we could use the same technology and implant it, but rather than trying to treat pain, we were trying to facilitate the initiation of locomotion. And we tested that in 4 subjects. We had a great team from multiple universities, and that we finally were able to test our first subject. And that was a subject that was motor complete. Now, we started with motor complete because that's the toughest one. That was the toughest challenge. And so I thought if a person is incomplete, they already have some movement, and how much they can recover varies quite a bit.
Mhm.
But if they're complete, we thought everyone knows that if 6 to 12 months after the complete injury that they have not recovered, they're not going to recover. No other adaptation is possible. So we took that model because rather than studying a large number of subjects to get to the appropriate statistics to show that this is real, we focused on individual subjects, and we shot our target was to do 4 subjects. But in the first subject, we learned so much. And it really, the subject with it was a single case subject, and we found out not only did the individual recover with the stimulation and the training, recover voluntary control of the lower legs, some control of the lower legs, and actually quite a bit of control, but also regain functions of other organ systems. And that was a real puzzle. We didn't anticipate that at all. But it turns out that that single observation has caused widespread application of these techniques to multiple organ systems. And the question is, how does this occur? We could get into that a little bit, but the fact that it occurred, again, this is a systems biology issue. No one ever expected that to occur, including us. In fact, we never did the control study of testing whether the person could voluntarily move. We knew he couldn't move, so we didn't make any measurements of it. But then when we found out he could move, then we were all shocked.
So I just want to, if I could jump in. So we have a complete cord transection, and you're applying the neuromodulation. How, I guess I can understand, like before I was thinking with central pattern generators that it was somewhat involuntary, but you're saying voluntary. They could somehow, there was like a connection from the brain down to the legs again, even though this is a complete transection.
All right, a couple of things. Number one, the human injury is almost never a complete transection like you would see surgically. What happens is several segments have been severely injured to the point that no information can get from the brain to the lower part.
Okay.
And the lower part needs to be intact. And that's where this central pattern generation set of neurons are located down there. And so if it's a thoracic or even cervical injury, that part of the spinal cord can largely be intact. And so I forgot where were we going? What was
I
it?
guess I guess I was thinking more in terms of it like literally being cut, and was wondering, well, how is any information getting across that gap? But it
Yeah,
seems like more of like a crush injury where there might be some functional
right.
fibers that are
Yeah.
passing some information.
We still, well, anyway, all the subjects has been studied since then with that kind of injury. Almost all of them have regained some voluntary control. In other words, we knew it was voluntary because a person could follow music, could follow other signals that tell it when he should be moving or try to move. So we knew it was voluntary. Now, there's a lot of controversy about what is really voluntary and what's automatic, but there's no need to get into that. We know that the brain is having some impact. In fact, we recently just reported in the neuroscience meeting that not only can this person, a person that's been injured over 11 years, can exert some positive control in initiating a movement, but he also can voluntarily inhibit some networks. And I don't think anyone had ever demonstrated that. Now, the importance of that is when you move, your brain sends not only excitatory signals, but it also sends inhibitory signals to muscles so you can get the inhibition and excitation of different muscles. And so that's pretty remarkable. I think that a person can regain the inhibitory side as well as the excitatory side of the movement.
And at this point in humans, those who are using this, is it just an internal electrical stimulation approach, or are there people doing the combo of that and pharmacological? Are there people using the external, on the skin type of electrical stimulation?
Okay, all right. That's a very good and logical question. But as soon as we saw this, we knew of the way of there was a special type of stimulator that was developed in Russia by a Russian colleague that we knew, and it was developed to enhance the performance of athletes. And we thought, and particularly Yuri Gerasimenko, he said, well, why don't we try this stimulation and just put it on the skin overlying the spine?
Hmm.
And when that was done, and all the experiments from the beginning has shown that, well, there's a lot of potential here. And then and at that point, we stopped doing implants because there's so many advantages to the transcutaneous stimulation. Not only do you eliminate the surgery, which is very costly and makes everything more complex, but you could use it for a short period of time or for a long period of time, and you could just remove the electrodes that you've placed on the skin like it would be EKG. And so there's so many advantages, and the technology is not so demanding, and therefore it could be used in many different situations, undeveloped countries, and so forth. It has so much more potential, and we are still testing that potential. Right now, our total focus is on transcutaneous neuromodulation, and the paper that we're just submitting is the paper that has shown pretty remarkable functions that can. It's hard to say that it will be equal to an implant, but if it's not equal, I think it can be pretty close because we're still trying to refine the techniques, and we don't think we're even close to optimizing the stimulation parameters that's necessary and better for a given subject at a given time for a given disorder.
Yeah, yeah. Because as I'd mentioned in the email, our in spinal muscular atrophy, which my children have, they're doing some small subject clinical trials of internal implant stimulation,
Mm.
and with some really exciting results. And I'm already thinking, as a physical therapist, could we maybe do some transcutaneous work? But I know, like with my kids, they're only 6, and anytime I have turned up the E-stim unit high enough to create an actual like muscle contraction, if you will. I
Mm.
know that this is more going at the sensory input to the spine, but they don't tolerate it well. And so
Yeah.
is that part of what kind of the optimization of the parameters with transcutaneous that y'all are working on? Is this working on tolerance, or is that not an issue with the waveforms that you're using?
It's a huge issue.
Yeah.
In fact, our our thinking and observations are that we don't want the muscle to contract when we stimulate. And because as soon as you start to stimulate the muscle directly, you're bypassing the automaticity that's built into the spinal cord already.
Yeah.
And so it can interfere with the recovery, with the adaptation and recovery. Now you can get some result, and it is helpful to go slightly above motor threshold to get the excitability level up, but you need to back off as much as you can from the level of stimulations to let the automaticity recover in these networks. So as long as you are going to maintain the stimulation, the person is always going to depend on that stimulation to have that function.
Mm.
So we think we can gradually withdraw the stimulation, and even to the point that we can withdraw it. How? We can withdraw it by giving less often. And for example, we're certain that in cerebral palsy, for example, we've worked on this quite a bit, and we know that we can gradually back off to an amazing level. Or it may be that you need the stimulation to get to a certain level, and then you can just not use it for a month or maybe 2 months. But then gradually you might lose some of that function and need a little more stimulation. But those are important questions that we don't have a good, fine answer for all of the different conditions. But these are the concepts that are important in trying to apply neuromodulation. And I keep emphasizing in the beginning, I make the analogy we're where the Wright brothers were when they did the first flight in understanding the potential of neuromodulation.
Well, that's exciting. Taking it a little more in the direction of neuron healing and regeneration, actual changes that go beyond the moment that you are imparting an electrical stimulation or a pharmacological neuromodulation. Can you talk to any evidence of actual, reconnections, obviously plasticity there, but true, like, have y'all looked and seen in animal models that there's regrowth, compared to controls, there's regrowth when you use neuromodulation and actual healing of neurons and regeneration?
I think there's reasonable evidence that there's regrowth of axons and connections, but our thinking is that there's some neurons that have survived, but they're not really functional in terms of transmitted action potentials.
Mhm.
But the other possibility is that, but we know there have been hundreds of experiments in animals and some evidence in humans. We know that axonal growth can go, and can occur.
Even beyond, like, say, 6 or 12 months of post injury?
Yeah, I don't think that is the critical issue. I think one technique is to implant substances that will provide a scaffolding that nerves can grow through. So there have been many studies tried by many people with different strategies that have working on that. But the things that we're seeing with the neuromodulation is perhaps not dependent on axonal growth through the lesion,
Hmm.
and it's certainly not dependent on axonal growth from the motor cortex all the way down to the lumbosacral.
Yeah.
So, for example, there are connections all the way along the spinal cord, referred to as the propriospinal system, that the information can get through multiple synapses and eventually get down to the lumbosacral. Now, it takes a longer time to get there, and we know that when the subjects are recovering some of that voluntary information capability, it takes them a second or 2 seconds, or takes them some time to think about it before the signal actually gets down to the point where it induces some kind of response.
Okay. And on that same note, do you feel like combining, and I know it's very early, as you said, the Wright brothers' first flight, but do you feel like ultimately combining neuromodulation with other therapeutics or developing therapeutics would have an additive effect, or could potentially, really take this a lot further than it currently is?
No question about it. No question. I've emphasized this from the beginning. As soon as we saw, even before we saw these latest results, anybody that's doing a clinical trial, they're implanting cells and trying to get regeneration from stem cells and so forth, or doing anything. If they do not take advantage of the adaptation that can occur with stimulation, for example, and pharmacological and electrical stimulation, and the training. If you're not doing that, you're missing the boat.
Wow.
And so you and a number of people have approached us as in trying to add this to the stem cell idea, and I really wanted to do this, but it makes the experiment so complicated
Yeah,
and more subjects. So no one has gotten there. But if anyone does not take advantage of the activity-dependent plasticity, they're missing the boat.
So on that note, we have some, I say we, it's not my experiments, but some experiments that I helped compel to occur starting later this year in mouse models of SMA, ALS, and, well, peripheral nerve injury for sure. We're hoping for a spinal cord injury as well. But earlier you mentioned the pharmacological stimulants were serotonergic. And interestingly, the ones that we are going to be using are also serotonergic. I can tell you more details about the actual method of application and how things are designed now, but it makes me wonder if if some sort of movement protocol or activity protocol should be added in order to give the best chance of some sort of regenerative, result, right? Or I mean, essentially, they're going to be an IP injection of these different drugs, and looking at, do we get reinnervation at the neuromuscular junctions, et cetera. And yeah, I wonder about that. I wonder if you would suggest strongly that there is some sort of movement component added to the protocol, added to the experiments. What are your thoughts with that?
There's no question about it. The more I look into this whole thing, the only way I can explain most of it is there is some very important activity-dependent mechanisms that are guiding the reorganization of these networks.
Mhm.
So if you don't activate them, there's nothing to tell them how to get reconnected.
Mhm. And in the experiments and animal models, say rats and mice, are we talking about simply putting them on a little treadmill, or are there really specific type of movements or things that you have them doing in these experiments?
Well, the most obvious thing is to use activities that are related to where the dysfunction is,
Mm.
but there's not very clean specific evidence of how specific it has to be. I think you can be too specific.
Hmm.
But I think if you're too specific, you're probably not activating all of the networks that actually contribute to a movement.
Yeah.
For example, in just going from sitting to standing or stepping, you're basically your nervous system is set up as soon as you have decided to step, your whole nervous system has set up to engage almost every muscle in your body.
Yeah. Yeah.
So it's an advantage. It's an advantage not to do real specific. I think we've done a number of experiments along this line, but you probably can be too general as well. But we just don't have really definitive information on just how specific it has to be. In our first experiment with cats, we tested this. After the spinal transection, we trained them to step, and they could step. And then we trained them to stand. And after we trained them to stand, and then we tested their ability to step, that most of them couldn't even step.
Because they got away from the stepping training and you were focused on standing?
We had activated a very specific set of networks that work for standing.
Huh.
But we were able to take that animal and then train it to step, and it learned to step.
Again.
Well, that animal had not been trained to step. It was an adult animal, and for 12 weeks it had never stepped.
Mm-hmm.
And we gave it a drug. We gave it strychnine IP, and in 30 minutes we put him back in the cage, and in 30 minutes we put it back on the treadmill, and the animal could step full weight bearing over a range of speeds.
Wow. That's interesting, IP, because I mean, in my mind, I was always thinking that you were infusing it around the area of the lesion,
Yeah.
but you're most of these pharmacological neuromodulators, you're just giving them IP.
Well, this was a gross experiment at that time. This was just the big test,
Mm.
but more specific location would probably be better because some of these drugs has multiple effects on multiple systems,
Mm.
and so you'd have to have such. In other words, we probably in humans, I don't think anyone would want to give enough strychnine
Yeah.
that it would you would have the side effects, which would be death.
Yeah. Yeah, well, I'm gonna have to. I'll definitely revisit with the researchers and see if we can figure out how to bring some movement, some activity into. So thank you for that. So,
But let me make, you mentioned one point. Let me just quickly state about the motor neurons and ALS, for example, you lose motor neurons. And none of this works if you don't have motor neurons, because the motor neurons are where the signals eventually have to go to individual muscles.
mhm.
So if you don't have the motor neurons to activate, you can have all the inner neurons, but they don't have the neurons that go to the muscle.
But if they,
So,
let's say, in the case of SMA, which has treatments that significantly slow or even stop the progression of the loss of motor neurons, as long as there are some remaining, then
mm,
neuromodulation might have a place in increasing muscle
yeah,
output and function. Yeah.
right. No, I think that's possible. Yeah.
And speaking of, you mentioned cerebral palsy, which I didn't know that you had used this in CP. That's exciting. What are all the different syndromes where neuromodulation has been looked at or is even maybe being used clinically at this point?
Uh, well, there's a recent podcast about 3 days on cerebral palsy and developmental diseases that occurred just a couple of months ago, and I gave one of the lectures. Uh, so there's a lot of interest, tremendous amount of interest in that area of developmental type of disorders. And I don't know what to say about this, but what we've seen, I'll just simply say, is amazing. The responsiveness of different kinds of disorders. It's what can happen in those kids is really exciting.
And has it, I mean, has it gotten into the clinic such that it's becoming part of standard of care, or is it still very much just in trials or experiments?
It's certainly not in clinical trials, and that's where we are trying to get there. And our limitation right now, this is so frustrating, is we don't have the money to go as fast as we know we could go.
Mm.
And we know that we can, not only we and a lot of the people that we're working with, is it's quickly becoming realized that there's something really special here about neuromodulation in kids.
And if you had all the money you needed, what would that path look like? What are the things that you'd like to do that you need the money for right now that you're not able
Uh,
to do?
we would need the people. It's primarily people, and train the people of what to do to and especially the therapists, because here again the actual activity that we use, again, activity is very important in the training, and that's the type of training is very important.
The actual training that you are doing with the subject, with the
Yeah,
child or
with
the patient.
very subject specific. Yeah.
Hmm.
And there's many types of disorders that fit into this category, and I can only say it looks bright.
That's fantastic. What are the other syndromes or categories that you have seen this being used on and with good results?
Well, we've seen results that's affecting vision, hearing, swallowing, speaking, walking, just those as an example.
And then as far as syndrome specific, whether it's, one-time neurotrauma or degenerative diseases, or could you?
These are the ones that we've looked at so far are disorders that occur during development, early development.
Okay, great. Well, I don't want to take more of your time. We've gone over the hour, and I deeply appreciate you taking the time and sharing your knowledge, and hopefully through this podcast we can get the word even further out about neuromodulation and utilizing it and across many contexts to help people with all sorts of ailments in this realm. And yeah, so if anyone wanted to follow your work or look more deeply into the papers that you've published or what you're working on right now, where would you point them?
We've published several papers in the area of cerebral palsy in Frontiers journals, in I don't even remember the name, but I would say that if you just look in PubMed, you will suddenly see a large increase in number of studies, pilot studies of upper arm function and walking and so forth.
Right.
So
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 searching
I
for
would
your name in PubMed would be a start. And then I'll certainly in the show notes I'll link to your website or webpage at UCLA. Are there any other web pages or websites that you would suggest?
Well, our lab right now is at the Rancho Research Institute, which is a part of the National Rehabilitation Center in Downey, California, which is, of course, focuses on rehabilitation techniques. And we've
Yeah.
initiated a lab there. The name of the lab is the Broccoli Impossible to Possible Lab. And so if you want to encourage anybody to send us some money, it will help us to go faster.
Well, that's, part of what I'm hoping to do here is be able to do things like that and spread the word and yeah, find the those with, motivations to make this go faster to help it to do so. So thank you again so much for your time. I deeply appreciate it.
Okay. All right. Thanks a lot for the opportunity.